Now I'm going to demonstrate how we use the RNA single strand constraint in XRNA.js. So first, of course, we're going to zoom into the portion of the scene that we want to edit, and we're going to select a single strand constraint. And now we can begin editing. The first thing we can do is you can either click and drag while you're in the edit menu, or you can use the middle mouse button to go ahead and select a region, or of course you can right click to select that same region. Note that we are providing a reference of what portion of the scene that we're editing here, and we can also go ahead and start editing our data. There is, as always, a color editor and a font editor if you wanted to recolor the portion of the scene, so I'll just show you that here. And we can make it red if we like. You can also change the orientation, so this constraint sort of keys off of a notion that single-stranded regions are sort of arced, and so they always lie along a circle. So you can make it clockwise, you can make it counterclockwise, or if you want, you can simply straighten it. You can also, while keeping it either clockwise or counterclockwise, you can change the displacement along the normal, and I understand that's a little confusing. What exactly does that mean? But what it means is we're taking the difference between these two bounding nucleotides, and there's implicit normal along the axis, and so it points either in this direction or the opposite direction. And so if you change this value, it pushes it out further away from these two bounding nucleotides, so it makes the arc larger essentially, and you can have a negative displacement if you want the arc to be really sort of flat. Okay, so that's the edit menu for the single-stranded region. Let's go ahead and format. Here we can see our base pairs editor, and this is pretty straightforward. It's essentially the same as a single nucleotided constraint because there's no real indication of how many nucleotide pairs you want to make if you're trying to create a helix, say between this single strand and this single strand. There's really no way to tell, and it's also unclear necessarily where you want to create the base pair to, so it's essentially populated the same way as a single nucleotided constraint, but you can have more than one base pair using this constraint, so that's a key difference. This is also available is the annotate menu. So if we go ahead and we start annotating in this region, we can, for the first time, we can go ahead and change the number of nucleotide index increments that we want. So we can make it 5 as an increment. So this means that every 5th nucleotide will be annotated. And we can change where we want that to start from. So we could say, by default, it's going to start at the lowest one, so that's 1581. But we can change it if we like to say 1582. So let's make it 1582. And now we don't really have enough base pairs for 5 to be applicable, so let's make it 3. Every 3rd nucleotide is going to receive an annotation. So we create annotations, and you can see just like that, every 3rd nucleotide becomes annotated. This has been the RNA single strand constraint in XRNA.js.